Hi, welcome to my channel. Today I'll explain mechanism of enzyme action. Catalysis is the prime function of enzyme and the chemical reaction in which reactant had to be in activated state or transition state. Enzymes have lower activation energy. Energy required by the reactant to undergo reaction is known as activation energy. Reactant, when heated, they attain the activation energy. Enzyme reduce the activation energy and the reaction proceed at lower temperature. Enzymes do not alter the equilibrium constant, only enhance the velocity of reaction. Role of enzyme is comparable with the tunnel made in a mountain to reduce a barrier. This is the graph of energy with the enzyme. This one is the energy, activation energy with the enzyme and activation energy without the enzyme. Energy change in a reaction. Here, enzyme lowers energy barrier of the reactant, thereby making the reactions to go faster. Enzyme reduce the activation energy of the reactant. All the biological systems occur at a body temperature below 40 degrees Celsius. Enzyme, first of all, enzyme substrate complex formation. The prime requisite for enzyme catalysis, that is a substrate and enzyme. Substrate must combine with enzyme at the active state to form enzyme substrate complex. Result in the product formation, that is, enzyme react with the substrate to form enzyme substrate complex and it cleave to form enzyme and the product. Next is the Lockandkey model or Fischer template theory. Theory was given by German biochemist Emil Fischer. Structure or conformation of the enzyme is rigid. Substrate fit to active site, just key fit into the proper lock or hand into the proper glove. That is the Lockandkey. If you want that I can explain this process in Hindi also, please to subscribe in the please to comment in the comment box. I would like to explain the same phenomena in the Hindi or any other language also. Pre-shaped template where only a specific substrate can bind. No scope for the flexible nature of the enzyme. Model totally failed to explain many facts of enzymatic reaction. That's why other theory will came to exist. That is a induced fit theory or Koshland model theory. Koshland in 1958 explained that more acceptable and realistic model for enzyme substrate complex formation. Active site is not rigid and pre-shaped. Essential features of the substrate binding site are present at the active site. Essential features of substrate binding site are present at the active site. Interaction of substrate with the enzyme induces a fit, that is, formation of a strong substrate binding site. Sufficient experimental evidence from the X-ray diffraction studies. From this study, we come to know that allosteric modulator and competitive inhibitions occur on the enzyme, and this is a figure that explains the induced fit phenomena. First, enzyme, they react with the substrate, and this one is the structure of this. First is by the Lockandkey. This is Lockandkey, and in the you can see that this structure of enzyme and the substrate, they are complementary to each other, or we can say that they can easily fit into this enzyme. And on the B figure, that is induced fit theory, here this structure is not similar but according to their change in the conformation, they can easily fit. Next is a substrate strain theory. Due to the strain is like energy interactions. Enzyme is substrate is fit into the enzyme. In the substrate strain theory, that is a new theory. This one is a new theory. The substrate strain theory, the substrate is strained due to induced conformation change in the enzyme. Here, substrate bind to the preformed active site. The enzyme induces a strain to the substrate, and the strained substrate leads to the formation of the product. Combination of the induced fit model and the substrate strain is operative in the enzyme action. That is the substrate strain theory. Or we can say that according to this theory, the substrate is strained due to the induced conformation change in the enzyme. Or we can say that the enzyme conformation induced to lead to the changes in the substrate structure, and substrate fit into the enzyme. If you really like this video, please do subscribe my channel and don't forget to like and share this video. And if you have any doubt regarding anything, please do comment in comment box. I would like to explain that topic in detail also. Thank you.